time we met here, we talked about light microscopes. And we said that microscopes were basically being used to observe very small organisms, right? And now we're going to talk about different ways that we can use our light microscopes. So this was our previous slide that we left off on. We were talking about a compound light microscope. We talked about how we can determine total magnification. We just did kind of a review. And now we're on to the different types of microscopy out there. Now, these three types that I'm going to talk about are based on the same microscope that we use in here in the lab. The difference is that they have special filters that can make them either a bright field, a dark field, or a phase contrast microscope. And we're going to look at what those differences are, okay? So if I'm using a microscope that has a bright field filter, and this is basically what we have in here. When we look at our slide specimen, what we see is that the slide specimen appears darker or has more contrast, while the background appears clear, right? This is what we've been seeing when we stain our cells. We see those positive stacks, for example, little purple circles, and then the background area is white. So this is really nice to observe specimens that are like naturally not stained, like let's say like pond water organisms, or specimens that have a natural color to them, like our pond water organisms that were containing chlorophyll that were green, or to observe stain samples. And that's why we use our microscopes that are set for bright field microscopy. Here are pictures of bright field microscopy, and hopefully these look familiar. These should look like things, I mean, maybe not the exact specimen, but the colors and the arrangement should look somewhat familiar. Now, we can put a special filter on our microscopes, and we can change our microscopes now to be something called dark field microscopy. This is basically the exact opposite. It's like an inverse. It makes things appear almost like they're a negative stain. It's just a filter. And so what this filter does is it makes the background dark, and now what's going to happen is my specimen will appear to be brighter. So it gives me a contrast. The beauty of this is that the light is reflecting back from my specimen, and it allows me to see things that might not stain well. I have certain organisms, for example, syphilis does not like to pick up stains. So when I try to look at syphilis in light field microscopy, because it doesn't stain well, it's hard to see it. I can use a dark field microscope though, and I can see the organism. That's actually syphilis there at the top, those little like snake-like things. My disadvantage though is any dust or debris on the slide is also going to reflect the light. So I get a lot of background noise. So I might get a lot of, just like little spots that appear. So this would be what dark field looks like. So again, we see syphilis here. It's kind of like little snakes. You can see at the top, those are some diatoms. So that's from pond water. Here we have a little nematode, a little worm. So you can kind of see, just by looking at them, that they're very bright while their background is dark. I'm not having to do any special staining. It's just a filter that I'm using. Now, the way that this works then is really just based on how the light is passing through that filter. When I'm using bright field, my light is hitting my specimen directly. When I'm using dark field, the light is hitting the specimen, but what's hitting my oculars is the reflected light. It's almost like looking at a shadow of somebody. Like if I shine a flashlight on them, I'm looking at their shadow more than them. Okay? So it just is dependent on which way the light is flowing. That's really the big difference. So some advantages. The reason we would want to use a dark field filter would be if we're trying to view cells, again, that are unstained or difficult to stain. If we have very thin, transparent cells. If we have cells that are, for example, like our pond water organisms that don't have a natural coloration to them, this would really help us. Now, with this, again, unfortunately, we do get a lot more background noise. So dark field microscopy is kind of an art, learning what you're supposed to be seeing as opposed to just the background. Okay, true or false? Dark field microscopy would be a good one to use. Okay. To view our pond water organisms. True or false? True. What do you guys think?
True.
True, yeah, it would be a really good one. We said unstained cells, right? So that would be a great one. Okay, this is a throwback. If a microscope has a resolving power of 0.3 nanometers, that means it can distinguish between 2 points that are how far apart?
0.3.
0.3, yeah. Doesn't that feel like a trick question? It's like it's too easy. Microbiologists hate math. We try to make everything easy. Our oculars, our tins, we try to make things make sense, all right? So 0.3 nanometers means I can distinguish points that are 0.3 nanometers apart or wider. Okay? True or false? Immersion oil is going to help with resolution. True, right? It's going to help us see our images more clearly and better. 0.3 nanometers can distinguish between 2 points that are 0.3 nanometers apart. Oh, it's just the same thing. So if I tell you something has this for a resolving pattern, then that means that that's the farthest it can, or the closest that those can be together. Yes, sir.
I have a question. This is my experience on previous series classes. When you have a case for sure which concepts and content, did you post like the study what you're gonna have on the
Yes, I will post
not just like past live, but it's it's just impossible to remember
Yes. So he just asked me, am I going to post a study guide for the exam that's coming up? The answer to that is yes. I will be getting your study guide sent. All right. So that way it'll help you kind of focus as you're studying through. All right. Because it is. It's a lot to remember. It's a lot. I know. Okay. Let's go to the next type of microscopy. This is a special one called phase contrast microscopy. This is another one that's good for observing organisms that are unstained. And what's going to happen here is that we're going to control how the light reaches our specimen, passes through the specimen, and then reaches our oculars. Basically, what's going to happen here is our specimen, if it's transparent, is going to appear either brighter or darker, depending on how we adjust the phase. You guys probably recognize this as iris diaphragm, right? This is what we already do. We have that special tool in our microscopes. Some don't. But the iris diaphragm is actually what is going to help us with phase contrast. It's going to help us adjust the light. The optics in the microscope are able to adjust for how much light we're allowing to get through to give us a different image. All right, so this is what our phase contrast will look like. What's going to happen then is the light's going to come through the specimen, and we're going to be able to control how much light is being on the specimen and how much light is being reflected from the specimen. It gives us more control. Now, the reason this is important is it allows us to actually kind of focus the light on specific details in the cell. Instead of the entire cell shining light back, we can make the contrast a little different so that we can see like just a nucleus or just some cell wall material. We can make it more focused. All right, so it allows us to kind of dial in. So let's look at the differences here. Here's a standard bright field microscope. Okay, so this is one that we would just be looking at here. We have an unstained cell. We put it on our microscope. This is what we would see. If we had a dark field filter, this is what we'd see. The background would be dark. The light would be reflecting from the specimen. Okay, so we see kind of like this outline, this halo. We can have our phase contrast though, which is like a combo of the two. Phase contrast, notice the background's a little lighter, so we're not blocking it completely. We're getting a little light shining off the cell, but we're also letting light pass through, so we're able to see a little bit more of a contrast or detail. So that's what our phase contrast is doing. And again, we do that with our iris diaphragm. That's the beauty of the iris diaphragm. So a lot of times when I'm walking around the room, I'll have somebody show me their stain and then I'll make a little adjustment with the iris diaphragm. They're like, how did you do that? What did you do? Because now it's like magic. Now I can see it. It's because I just adjusted how your light is directing through. Okay, so don't be afraid to play with that iris diaphragm. Here's some more pictures that you can see where we've adjusted with the iris diaphragm to see all the little cells and their neat little structures. Now, the advantage of this is because we can adjust the light, we can look at living cells, things like pond water organisms, and still see them. We're able to do that, right? We don't have to fix our slides. For example, our wet mounts weren't heated or fixed, so we can look at things that are living. we end up with cells that don't need to be stained, but we still get a nice high contrast. We actually even use it in our lab on stained cells to give us a better contrast, because there's parts of the cell that doesn't absorb the stain as well. So then we can really see things like the cytoplasm and stuff. Now, if we wanted to go another route to look at cells, we would have to get a whole new microscope. So this is the fluorescent microscopy or microscope. And we don't have these in our lab. They are another type of microscope completely, because they have special filters that are very different than what the light microscopes are that we use. What we just talked about with the phase contrast and the light and dark field, those are all just filters that can go on the same microscope. When we get to fluorescent microscopy, it's a whole new tool, whole new microscope. Fluorescent microscopy is going to be used primarily in hospital settings or in research labs. And what they do in fluorescent microscopy is they use special dye on their cells that tags the organisms with a special fluorescent molecule referred to as a fluorochrome. Basically, it's kind of like how we dye our cells. You know how we use like methylene blue to dye? Same idea, except they're working in a dark room. They're dyeing with a fluorochrome that is going to fluoresce when light hits it. This is similar to what we do when we look for things like cancer cells. We use different types of tagged material. It can be like tagged iodines. We can look for things that will tag to sugars in the cells. So what's going to happen is these fluorochromes or these tagged molecules can go through our body and then they attach to cells. And then we can look at you under a scanner and we can see where we see fluorescence. Same thing is true here. We're just doing it on a slide. So we're just looking for fluorescence. So here's an example of a tumor that has been treated with fluorochromes. And so what we can see is that the cells have absorbed the fluorescent color. When I look at them under the microscope excites the fluorescent color and it glows. So any cells that absorb this color are going to glow or appear very beautiful to look at. The nice thing about this, it gives me a 3D view. I can look at living or dead cells, whichever one I want, and it's a nice contrast or resolution view. Again, this can also be used as a diagnostic tool. I can use markers, as I said, specific for cancers, and then only my cancers will fluoresce. So it's a nice diagnostic tool. As you can imagine, it's pricey, which is why we really only use it mostly in medical labs or in research labs. Okay, so to put this all together, our light microscopes that we use in here work great, but they're limited by their ability to do high magnification. Remember, if we go too high, we lose our resolution, just like if we blow up a picture too big. Our microscopes in here can go up to 1,000 times. Some microscopes on the market that we can purchase could actually go up to about 2,000 times, meaning we're magnifying 2,000 times. Those, as you can imagine, are more expensive. We find that the 1,000 times work well for us and they're affordable. Our resolving power in here is 0.2 microns or 0.2 nanometers, which means that we are able to see our specimens and see them clearly. But if we really wanted to see individual detail, we would really have to go to a stronger setting. Because of this, in here, if we tried to view viruses, we wouldn't see anything. They're too small. If we would try to view mitochondria or certain organelles in a cell, we wouldn't see anything. They're too tiny. To deal with very small structures, we need to go up to the next big tool, and that's the electron microscope. The electron microscope has a resolution that'll get us up to about 2 million times. That's pretty darn good. This is what your electron microscope looks like. Isn't that just an odd microscope? You're looking at a computer screen, and your sample is inside this large machine. The way the electron microscope works is it's going to basically be exciting electrons within the cells. And those electrons start to kind of bounce around and move, which starts to generate kind of a 3D image. So we can kind of start to see what our cells look like. Now, there are different types of electron microscopy. We can use what's called TEM, T-E-M, transmission electron microscopy, which will show you internal structures of a cell. Now, the magnification here is about 100,000 times. And it gives you a 2D image. And let me explain why. You basically take your sample, the cell that you're going to look at, and your microscope is going to slice that sample into little tiny images. Kind of like LiDAR. Have you guys seen LiDAR where they do scans of the topography of the land and they can take little images like slices? That's what happens here. We get little slices. So we get a 2D view. I see the organism, but I see them kind of cut in half, basically. So it's a nice two-dimensional view. The other thing is to create these thin slices, I have to use a special process. And what that actually does is it dehydrates and kills the cell. So when I do this, I'm killing the organism. And because of the fact that I'm killing it, that can lead to what we refer to as artifacts. That means that maybe the cell's changed a little bit. Okay, so it can lead to things where maybe the cell shrunk a little bit, or it doesn't quite look the same. Here's a picture of a pond water organism through a TEM microscope and a pond water organism through a light microscope. So this is what we would see if we were looking at this euglena, for example. That's the organism. We would see just kind of like darkness and a little contrast. If I put them under a scanning electron microscope, this is what I see. And I can tell this is a TM image or a TEM image because it's a two-dimensional cut in half image. All right. Now, the other type of electron microscopy that I can use is something that's newer. It's referred to as SIM technology, which is the scanning electron microscope. The way this works is we actually take our specimen and we dust them with a heavy metal. So let's say we have pond water organisms, we add heavy metal, usually we use a gold dust, over the cells. And then when we excite that cell in the electron microscope, the gold is actually what starts to vibrate and reflect the image back. But everywhere that that gold has coated is what gives us that image. Okay, so it's like I'm dusting you in gold and it's shining a light at you and I'm seeing the glimmer of the gold, more so than the cell itself. The beauty of this, I get a higher magnification and I can get a 3D image. So now, here's that same little euglena I was looking at, but now look, I can see a little bit more detail. See how that works? It's not like it's just cut in half. I can actually see it. So the difference here also is I'm not killing the organism. Dusting them with gold doesn't hurt them. So depending on what pictures I want to take, I can use either that TEM or that SIM technology. It's up to me as to which ones I want to use. The last few terms we want to know for microscopes are ones that we use in here, things like parfocal. That's a term that we've been throwing out this semester. Remember, parfocal is the ability for our microscopes to stay in focus as we move from the 4x to the 10x, the 10 to the 40, and the 40 to 100. It's not perfect. We always have to do a few little adjustments, but at least we're not having to do a big horse knob adjustment and trying to find stuff at every single level. Parfocal basically means that once we get something in view, it should stay in view. Now, one thing I do want to let you guys know is as we keep moving through our class, remember we're viewing bacteria. So when you're viewing things at that 4, 10, and 40x, you're really not going to see the cells. They're still too small. We can see like the dye, but we have to go to the 100x to actually see the cells. So when I walk around the room, if I'm looking at somebody at 40x, it looks like I said, like a Jackson Pollock painting. There's like splashes of dye everywhere. But I can't see anything yet, okay? I really have to go to that 100x to see the detail. So that's why I'm always like, yeah, go to oil, let's look at it. The other thing is the idea of field of view. We brought this up with our letter E. I run into issues where when I'm at a lower magnification, I get a bigger field of view. This is the other thing that's nice. When I'm at the 4 and the 10x, that's the time to scan around and make sure I have a good view. Even though I can't see the cells, I can tell if my dye is too clumped or too thick. That's why a lot of times when I walk around the room, I'll move you to an area where it's a little thinner so you can actually see the cells. This is because we're able to see more of a broader view. As soon as I go to that 40 or the 100, I'm dialed in. I'm getting in close, and it's much harder to scan now. It's like, I went from this broad view to, whoop, I'm like right here, okay? And that's nice, but remember, just like what we saw with our letter E, we can get so close we lose details. Right, so you can get like too close. It's like doing a close-up and now somebody's like right here. It's like, that's a little too close. Maybe fix that out. Oops. The other thing we can do to view our organisms, which we've been doing in here, is to do staining. So as we've seen with our pond water organisms, natural cells really don't have a lot of color to them. If we were to look at like our cheek cells, they don't really have color. So when we dye them, the reason we do that is to give them more contrast. So here's a picture, for example, of bacillus that is undyed. Not very exciting. Here's a picture of bacillus with crystal violet. Isn't that nicer? So much nicer. We can actually see the cells. This is the reason why we stain our cells. So we want to be able to see the organisms more clearly. As we've said in our lab book, we have different kinds of stains that we can use. We have basic dyes that are attracted to the cells because our cells are negative. We have acidic dyes that are repelled by those negative cells. So they will stain the background of the slide. We saw that when we looked at our cheek cells, for example, we did a negative stain. We can use mordants. And remember, a mordant is something that helps increase the stain intensity. So the mordant is going to make your stain color better, your stain contrast better. And we can use simple or complex stains. Simple means we're using one dye, complex means we're using two. This is all familiar to us because it's a nice review from lab, right? It's kind of nice. It's like, oh, you kind of relax on this. We can do our simple staining with something like crystal violet, methylene blue, carbon fusion. These are all basic stains. And they will stain the cell, and the cell itself will pick up a nice color, not staining the background. Here's another simple stain that we can use. We can use a negative stain, and that's still a simple stain because it's using one dye. In this case, though, we're staining the background. So we can have simple stains and then a simple negative stain. And then these are the ones that we're doing now. Our complex stains, we graduated. We learned about our gram stain the other day, right? Our gram stain, as I said, is the cornerstone of microbiology. It tells us which road we're going to go down when it comes to a diagnostic tool. Today, we're going to learn about acid-fast stains. Acid-fast stains are used for a few genera. There's only a couple out there that are acid-fast. One of them is Mycobacterium. And Mycobacterium is an organism that causes tuberculosis. You heard of TB? Sometimes called the white plague. It is a tuberculosis strain, and we still have TB around. One of the ways you can see if a patient has TB is to do an acid-fast stain on a sputum sample. We also can look for structures in our complex and differential stains. We can look to see if our cell can make things like spores, or if they have flagella, or if they have capsules. We're going to look at pictures of these. Our gram stain is going to be, remember, based on the cell wall. So the peptidoglycan layer in the cell, whether it's thick or thin, is going to help dictate whether our cell is gram positive or gram negative. So we can classify and separate our cells. Which one has the thick wall, gram positive or gram negative?
Positive.
Excellent. Gram positive. Yeah, gram positives have those thick cell walls. Gram negatives are the teenagers. They have the thin cell walls and they don't care. All right, they're like, whatever, take it, I don't care. So here we see our gram positive with this thick cell wall. The gram negatives, they have this very teeny tiny thin cell wall. What's interesting is to help them compensate for that thin cell wall, they also have something called an outer membrane. And we'll talk about that when we get to chapter 4. Basically, they've adapted an additional layer to help protect them since their cell wall is so tiny. They built another layer on top. As we know with our gram stain, it's very straightforward. We put our primary stain on, which is our crystal violet. We add our mordant, which is our Gram's iodine. We do a decolorizing step, and then we do a counter stain with the safranin. When we're done, this is what our stain should look like. Nice and purple for positives, nice and pink for negatives. Now, the limitation of the gram stain is that not all organisms can readily absorb those stains. For example, one of the ways that we find acid-fast bacteria is a lot of times when we run a patient sample, maybe we have a sputum sample, and we're doing a gram stain on it, and we see that the cells don't stain well. That's an indicator that those cells have something odd going on with their walls. which could be that maybe they have these special structures called mycolic acids that we'll talk about today. So the gram stain is something that works great for most of the cells. But if there's something odd going on, like an acid-fast cell, it won't be so great. The other thing we run into is age. And I mentioned this in your lab book. Does anybody remember why we need young cultures, less than 24 hours? Yes?
The cell wall will deteriorate.
Yes.
The
Cell wall.
will stain.
Excellent. And so what will happen? If a cell wall deteriorates, what's that going to do? Is that going to give us a false reading? Yeah. If our cell wall is falling apart, our gram positives now look like a gram negative. Excellent. The one we're going to do today is probably one of the prettiest stains. It's one of the stinkiest ones too. It just gets the dye smell. Okay. It's called an acid-fast stain. Isn't this gorgeous? I mean, this is again like a Jackson Pollock painting, but it's like beautiful. It's like Jackson Pollock on LSD. It's like, woo, that's kind of pretty, right? Okay, and no, I don't know what LSD is like. I'm just saying, basically. Okay, here we go. I know everybody's like, has she done this? Okay, so here we go. So the idea behind the acid-fast stain is we have a special cell wall. Their cell wall, to be an acid-fast bacterium, is one that is high in a lipid count, high in fats. So because of that, just like if I try to paint a candle, my paint won't stick to it. The same is true if I try to do a regular gram stain. It's not going to work, okay? The way I can get stains into acid-fast cells is I have to melt them, just like what I do with a candle. I melt a candle, right? It turns into liquid. Same idea here. I melt the cells, and not really melting the cells, I melt their cell walls, and now the dye can penetrate. All right, so that's what we're going to do today. So we're going to be melting cells with our Bunsen burners. Yes?
And that doesn't kill the bacteria.
It does kill them, which is okay. We want them dead. We don't want to have anything alive on our slides, especially with these organisms that we're working with today. The way we're going to do that is with our Bunsen burners. Today, we're going to be holding our Bunsen burners. Like, talk about a whole new level of stress. You know, it's so funny. There's another stain that does this too. And today everybody's like, oh, like nervous with holding the Bunsen burner. And then when we get to the next stain where they do it, they're just like, whatever. They just like pick them up, flip them over. It's like there's no fear. You know, like once we get past that initial fear, we're like, whatever. Okay, let's do a checkpoint. I am staining a patient's sputum. Okay, it is a rapid, inexpensive way to diagnose TB. What color would my bacterial cells appear if the patient has TB with an acid-fast stain? Let's see. In the corner. I didn't call any last time.
And the purple reddish?
Yes, that reddish color. Thank you. Yeah. So if we look here, it's like a magenta pink or a reddish color. Excellent. And then my background cells are going to be that blue color. Excellent. What color would a gram-negative cell be after a gram stain?
Gram
Pink. Pink. Excellent. Yeah. So we have our negative pinks. Now let's talk really briefly about some special structural stains. Special structural stains are going to be ones that are able to color specific parts of a cell. So there are certain organisms out there that are able to grow or create additional components to help them survive. These would be things like a capsule, an endospore, and a flagella. So we're going to talk about these things and how we would stain for them. A capsule, which we see here at the top, is like an additional shield that surrounds a cell. We encounter capsules sometimes in the organism that causes pneumonia. Streptococcus pneumoniae is the name of the organism, and we'll talk about it a little bit more in the lab. Streptococcus pneumoniae creates these additional shields around it. And so when I try to give a patient antibiotics, what's going to happen is those antibiotics hit that shield and bounce off. This is why sometimes patients who have pneumonia will give them an antibiotic treatment and they don't recover or respond. And that tells us we're probably dealing with a capsulated bacterium. So then what we can do is we can change the antibiotic to give them a combo that breaks the capsule open and then penetrates the cell. So this is why I've had students tell me, yeah, I had pneumonia and I took a round of antibiotics, it did nothing, and I had to go back and take another round with different antibiotics. It's because they were capsulated. So it's just an extra shield. This increases something we refer to as virulence. Virulence is the ability to cause a disease. So if I have an organism with a high virulence, that means it is capable of causing disease easily. If I have an organism with a low virulence, it's not able to easily cause disease. Our organisms that we work with here, within here, are all low virulence. So that's why I don't worry about us working with them. These ones are very difficult to stain. Because of those capsules, it's hard to be able to get the dye to penetrate. So we have to do a couple of tricks to be able to get our capsule colors to get through. We'll do capsule stains in here next time. The other one that we can look for are endospores. Endospores are basically almost like little eggs that are left behind. Remember Clostridium difficile? I mentioned that it had spores. This is what they would look like, these little green spores. The endospore stain is looking for these little spore structures that only some bacteria can make. Not all bacteria can actually grow endospores, only a few, only a special type. We see them in gram-positive bacilli. Staph, they can't do this. They're circulus, they can't do it. Poxy can't do it. So when we look at a rod-shaped cell, some of them can, but only if they're gram-positive. Okay? Now, the cool thing about the endospore stain is, number one, it's beautiful to look at. It's another one that we're going to do in here that has heat and wall. But the other thing is these spores are really resilient. We've actually pulled spores off of Egyptian mummy wrappings. And we were able to grow them and see what the bacteria looked like during the ancient times. Isn't that cool? Do you know how they knew they came from the Egyptian mummies? Because when they looked at them under the scope, they walked like an Egyptian. You guys are just looking sleepy. I gotta wake you. Okay. The other one are the flagella stains. We don't do these in here. They are heartbreaking. Okay? The way a flagella stain works is we're looking to see if our cell has the ability to move. And so what you do is you add dye to your cell and let it dry, and then you add more dye and let it dry, and then you add more dye and let it dry. You're coating the flagella. That's what you're trying to do. There are these little tiny structures. And as you coat that flagella, the goal is to get enough dye on there to coat it to where it's visible, because usually these are invisible. But we can't put too much dye on there because then the flagella gets too heavy, and what do you think happens to it? Yeah, it breaks off. Yeah, if I put too much on here, it's like putting a weight, and it breaks. It is a heartbreaking stain. We don't do it anymore. It was so hard. I have my students working so hard, and they're patiently waiting to dry in between every single step, and then they would go and look, and there'd be no flagella. It was just, there was no love for that stain, so we got rid of it. So on our flagella stain, what I do instead is I put out prepared slides. Much easier. There's no heartbreak, but we can see the flagella. Again, they just look like little hair-like extensions around the cells. We use them in microbiology to also help determine what kind of organism we're looking at, because there's actually flagella patterns. For example, you can have polar flagella, which means they're at the end. You can have like flagella on a bad hair day. You see that one, how it's like everywhere? We call that paratrichus. It means the flagella are just like everywhere. Okay, what color are gram-positive bacteria after a gram stain?
Purple.
Purple, excellent. What color are capsules after a capsule stain? That was a little tricky, huh? Let's give it a look at those capsules. I heard it. Are they glowing? They're not so much glowing, but they're clear. So they are colorless. The color can't penetrate. Oh, such a tricky teacher, huh? It's so tricky. So the color of the capsules is clear. There's nothing there, right? Because the color cannot penetrate. That's what I really want you to know there. Anybody know what kind of stain this is, based on what we've been seeing? The acid fast. Excellent. The acid fast ones. These are our mycobacteriums, our TB causing cells. And then the blue is the background. Excellent. So it's nice sometimes to look at the stain and be like, oh yeah, I can see what this is. I can tell what stain I'm looking at based on the colorations. Okay, that gets us through our microscopy. That's probably the one that students tell me is the most boring chapter, which breaks my heart, but I understand. And now we're going to jump on to cells. All right? So we're going to start our cells. So wait for a minute before you can switch your notes. What is that? Are those notes to be good in? Or no? I thought they were. Oh, are they not? Okay.
They're on
It's okay.
canvas.
If you don't have, I was like all of a sudden like, did my canvas switch? Because that's happened before. If you don't have them, don't worry. Just put page 1 and put it in missing words. And don't worry, you can still take notes. Don't panic if you don't have them. Just put, page 1 and put in the yellow words, and then you can fill them in later. So don't panic. Or what I'll do today, since I think I let him, I put him up yesterday morning. That's still kind of close. If you want to even just take pictures, that's fine too. Usually I tell you can't do that, but I will let you do that today because of the fact that that was kind of short. So I want to make sure everybody can do that. Okay, so in this chapter, we're going to learn a little bit about how antibiotics work. We're going to learn, for example, how penicillin was used as a miracle drug to be able to treat staph infections. That was amazing to us. That was our very first antibiotic. Nowadays, it doesn't work at all on the organism it was originally designed for. Remember, penicillin was found to have killed Staphylococcus aureus, right? Nowadays, if I put Staphylococcus aureus and penicillin together, the staph laughs at it and grows over it. Because it has been overexposed so much, it's built resistance. So we're starting to see that with a lot of our antibiotics. The bacteria, if we expose them long enough, they're like, I can figure out a way around this. Because bacteria are very smart, unfortunately. So in this chapter, we're going to start with talking about our cells. And as we've said before, cells are the basic unit of life. And what's interesting is that our cells can differ from one organism to the next. And again, if you need to take pictures, please feel free today, don't worry. The cells can be specialized for the areas that they're covering inside the organism. We'll have kidney cells that operate differently than heart cells, for example. And they can be complex. They can have different structures depending on where they're located. Now, we now agree that biogenesis is the basis of life, right, where cells are going to come from other life. And we refer to that basis as the cell theory. And that was invented really by a scientist named Virchow, who followed Pasteur, kind of put together all of the information that we learned about life coming from other life, and basically came up with a set of rules that the science group agreed with. And this is his cell theory that he came up with. He said all organisms are composed of one or more cells. Okay, that's true, right? All organisms out there, like whether they're bacteria or whether they're us, are composed of cells. The cell itself is the smallest living unit. We can break our cells down to the cellular level. That's as low as we can go. And cells come into existence only from other cells. So, for example, when we create a baby, we have a sperm cell and an egg cell unite to create an embryo. It's not like that embryo just boom appeared, right? It was because of two cells coming together. All right, so cells can give life or create other cells. All right. The other thing about cells is that they are all very small. Remember, Robert Koch was one of the ones who described the first cell. And he showed pictures of cork. They're very small units, right? And that means that we have to look at them under a microscope. There's a few cells out there that are large. For example, there's one cell in the human body that we can see without a microscope. Does anybody know what it is?
Egg cell.
The egg cell. Excellent. The egg cell is about the size of a period in New Times font. If you're typing up a size 12 New Times font, and you hit a period and you print out that paper, that's about the size of our egg cell. Isn't that cool? You'll never look at a printed paper the same way again, right? You'll see a period, you'll be like, oh, there's an egg cell. All right. So remember, we had Koch that was able to look at our cork cells. He was the one that termed that use of cells. But again, they're very small. The reason the cells tend to be small is if they get too big, it's hard to control them, okay? I can give you an example. Let's say I'm here in the classroom and I'm yelling out information like I usually am. You guys are close to me, so you can hear that information. If I'm in a small cell and my DNA is giving directions, I can hear it because it's small. Now let's say we go out to the track and I'm standing in the middle of the grass field and you're way in the bleachers. And I start trying to yell out information. Or I go to a stadium and I try to yell out information. So now we're in a bigger area. Are you going to be able to hear as easily? Not if I don't have a PA system, right? You're not going to hear anything. So that's kind of the idea here. As cells get too big, they get disordered. They can't follow directions. They become more difficult to control. We also run into problems where they can't get rid of waste as efficiently. Here, there's a trash can at the end of each bench. Whatever I don't need, I throw away. If I go to a stadium, I might have to walk a while to find a trash can. The same is true with cells. They can get rid of waste and they're small, easier. Now, the one thing about cells then, with them being so small, is we have to, of course, use our tool that we just learned about in the last chapter, our microscopes, to be able to see them. And we were really limited by our abilities to visualize the cells in regards to understanding them. It's easier to understand what you can see. So as our microscopes improved, our understanding and knowledge of the cell improved as well. They went hand in hand. So, for example, you guys saw this little critter. Here's E. coli. Remember how tiny it is? Yeah, E. coli is small, isn't it? And so E. coli, under a microscope, is still pretty darn small. It's like kind of these little pink rods that are like little clouds in the background. So even with magnification, these guys are still tiny. Now there's two major types of cells out there. There's prokaryotic and there's eukaryotic. On our quiz, I asked you about the main domains, and one of the domains is eukarya. I had a lot of people put eukaryotic. Eukaryotic is a type of cell. So I want to be careful. Eukarya is a domain. I want to be careful with that. These cells are really going to differ in how their DNA is housed. In eukaryotic cells, our DNA is housed in our nucleus. In prokaryotic cells, they don't have a nucleus. So their DNA is not housed within a membrane. So really that's their big difference, is where their DNA is housed. The other difference that we run into is what's inside of the cell. Our cells are bigger, so we have more structures present. Bacteria cells are simpler. They hardly have anything present. So let's look a little bit at these guys. They're going to be chemically similar in the respect that they require lipids, and they require carbs, and they require protein. So they can be chemically similar based on what their requirements are. Where they differ is in their cell wall structure. First of all, let's think about humans. Human cells, our cells are very flexible to go through our body. Do humans have cell walls? No. There was a question in your lab book saying, how would your cells stain with a gram stain? Do you guys see that question?
Yes.
Yeah. So the question you need to ask yourself is, do we have cell walls to hold in that primary stain? No. So if we did a gram stain on human cells, they would look gram negative. They would be pink because we're not able to hold in that primary stain. Isn't that cool? Because we don't have cell walls. We don't have rigid cells. Bacteria, they don't have any protection. They're out in the environment. They're out living around. They have these rigid cell walls. And those act as like little protective units for them. So we require the same things in regards to like carbs, lipids, proteins, DNA, RNA. But how we use them and what we build with them is different. Okay? So the prokaryotes, their name means before the nucleus. Pro means before, karyote means nut. Nut means nucleus. Okay? So this is before they had a nut. Because when the early scientists saw cells with a nuclei, they said it kind of looks like there's a little peanut inside. So that's why we got prokaryote. So before there was a nut. These are going to include our bacteria as well as our archaea. So both of these domains are going to be our prokaryotes. As I mentioned, they don't have a nucleus. Instead, what they have is a structure referred to as a nucleoid. What that means is their DNA kind of stays in the center part of the cell, but there's no membrane wrapped around it. That's the difference. The nucleus, think of it like a plastic bag. It's like a membrane wrapped around the DNA. The bacteria don't have that. They have the nucleoli, which are some proteins that kind of hold the DNA in place, but again, there's no bag wrap. They're very small sized. Usually when we're looking at prokaryotes and archaea, they're single cells. We haven't found any others that are not single celled. And a lot of them don't require oxygen to survive. So they can get different types of metabolism. They can use, for example, different types of carbons as opposed to oxygen to be able to live. So that term prokaryote then, again meaning before the nut, refers to the fact that the DNA is not enclosed. Eukaryote, when you hear eu, that means with. Eu means with. So eukaryote means it has a nut present or a nucleus present. And so our DNA is carefully wrapped in that sac-like structure. Our DNA is very protective of the cell. This actually makes sense because our DNA is pretty complex. A bacterial cell, their DNA is pretty simple. So it makes sense that as we evolved, we got more protective of our DNA as we started having to carry more information. As you can imagine, 30 trillion cells is a lot harder to keep track of than just a single cell. So we have 30 trillion bacteria or just singles. So when you're studying for your exam, one of the things I recommend is just to take like a piece of paper and jot down little like main bullet points. And I also recommend doing tables. Tables a lot of times just help you organize and synchronize a lot of information. When you look at my slides, you'll see that I have a lot of information repeated. For example, ATP, I had 12 different slides that show you ATP. You can jot down ATP in one line. Okay, so when you're studying, don't be afraid to kind of synchronize your notes. This is a nice table showing the differences between a prokaryote and a eukaryote. Prokaryotes, very simplified DNA, very simple circular type of DNA. Our DNA is paired. What that means is we get a set of DNA from our mothers and a set of DNA from our fathers. So our DNA comes from a combo of an egg and sperm, so it's a paired DNA. We, because our DNA is more complex, use special proteins called histones that wrap around the DNA. Think of this like a spool of thread. I have a spool and I wrap the thread around to organize it. That's what a histone is. It's a protein that the DNA wraps around to stay organized. Okay? If I didn't have that spool of thread, my thread would be everywhere and be tangled. So it's nice to have a protein that I can keep my DNA organized with. That's a histone. Prokaryotes, they don't have that. Their DNA is too simple. They don't need that. We're going to have specialized structures referred to organelles. And the organelles are basically little units within the cells that have a special job function. For example, we have an organelle that gives us energy. We have an organelle that recycles. They're like little apps that are inside the cell that do things for us. Prokaryotes are so simple, they don't need an organelle. They're really simple, small, they don't have to take care of other cells, so they can just kind of function on their own. They have peptidoglycan layers if they are bacteria. They have what's called a pseudomurine layer if they are an archaea. That's the difference between those two, different types of cell walls. That's why we separated the archaea and the bacteria. If we have cell walls present, things like plants, for example, have cell walls, they have a polysaccharide, meaning a mini sugar. Okay? So they're going to have a different type of cell wall as compared to our single-celled organisms. And then as far as reproduction, these guys do something really simple called binary fission. They basically go whoop and split in two. We do a complicated process called mitosis, which takes a lot more, time, energy, and effort. So again, we can see the size difference, but look at the complexity. I mean, these little guys, all they have inside of them, cytoplasm, DNA, they have ribosomes to make some proteins, and usually that's it. They're good to go. They might have a few other structures that we'll talk about, but pretty simple. Look at us. We've got all these organelles. We have this nucleus. We have recycling centers. We have a lot of things inside of our cells. So we have to do a lot more work because we're more complex. So let's keep exploring those prokaryotes. So if I looked at a prokaryotic cell, the first thing I would notice is that I have this plasma membrane. And the plasma membrane is basically a barrier to control what can enter and leave the cell. Think of a plasma membrane as basically like a bouncer. We don't have cell walls, but we do have plasma membranes. So pretty much every cell out there is going to have a plasma membrane. And again, it's like a bouncer layer that controls what comes in and what leaves the cells. We don't want things going willy-nilly with our cells. We don't want things coming in and out unregulated. Some bacteria, they even go a step further, and on top of that plasma membrane, they'll have a cell wall, as we mentioned before. And then some go a step further, and like the gram-negatives, they build another layer because their cell wall is thin. And then some can go even further and build a capsule. My goodness, it's like a fortress. No wonder we have a hard time killing bacteria, right? They have figured out ways to protect themselves. And we're kind of at their whims as to how we can penetrate those fortresses and get to the cells to kill them. This is why we're always changing our technology and the way that we approach killing bacteria. Yeah.
Is that like how we're dealing with AIDS and cancer? Is that how it can happen?
So AIDS is a little different, it's a virus. And so the way we're able to deal with AIDS is to be able to replicate the HIV virus which leads to AIDS. The HIV virus has to create a special enzyme. We're able to block that enzyme. That's the way we're dealing with HIV right now. So we prevent it from going to AIDS by keeping an enzyme away. That shows you how important enzymes are. Just by blocking one enzyme, I can stop somebody's HIV from advancing. With cancers, we usually are focusing on mitosis. Mitosis is cell division. Cancer is unregulated cell division. It's where a cell basically has its accelerator step. Picture like the accelerator of your car, it's stuck. It's like, what was it, the cars that were having problems with the Prius? That was having problems with the accelerator? It was Toyota, I remember. Which one was it?
I thought it was Toyota.
It was a Toyota, yeah. There was, a few years ago, they had to do a big recall because there was a Toyota car, I think it was a Prius, that the accelerator would get stuck on. Can you imagine, you're like driving and all of a sudden you're like stuck? That's my excuse if I ever get pulled over by CHP. It was stuck. They're like, you're in a Honda. No, it was still stuck. I swear, it got infected by a Prius. I was by a Prius. It infected me. Okay, maybe it might not. Anyway, the idea then is with cancer cells that start by to reproduce and to keep going through mitosis get stuck, so it keeps spreading. So if cancer goes through mitosis, it's I'm assuming it's a slower pace than like binary division, right? Correct. So if cancer tends to spread very rapidly, what happens to that very mitosis? Does it speed up? Great question. So cancers tend to all be slow and progressive. This is why we try to do screening. When you have an aggressive cancer, that means that it's not resting at all in between the mitosis. So it's replicating and then within 24 hours replicating, within 24 hours replicating. Where we run into problems when cancer does something called metastasize. What that means is it gets into the lymph or the blood system and it spreads and travels. And then those cells, wherever they land, they start to also go through mitosis. So now I have mitosis here and here at the original source. That's when we get a fast-acting cancer. Because I now have mitosis occurring in multiple sources. Great question. I love these questions. Okay, so, of course, bacteria are hard to kill because we have all of these protective things in place. Let's talk about what they have, though, inside the cell, because what we're going to find is a lot of their structures are similar to ours. For example, we said they have a plasma membrane, well, so do we. They have cytoplasm. That's kind of like an egg-like consistency, like a little jelly-filled consistency inside the cell. We have that, too. We have cytoplasm. It's kind of like this jelly-like fluid that holds everything in place. They also have ribosomes. We have ribosomes, too. Ribosomes are where we make proteins. Where we are a little fortunate is their ribosomes are different than ours. They have a different construction to them. We can target their ribosomes with antibiotics. So one of the main ways we can kill bacteria is to take out their ribosomes, which means they can't make proteins anymore. So then they can't make enzymes, and then they can't make cell walls. We can knock them out. Thankfully, because their ribosomes are different than ours, it doesn't hurt our cells. So that's a good thing. We mentioned cancer. One of the reasons why cancer treatments are so harmful, we hear about things like chemotherapy, right, which is harmful. If you ever know anybody going, my aunt right now has stage 4 breast cancer. She's going through chemo. And it is heartbreaking to watch her go through this. And it's one of those things, though, where you're poisoning your own body. That's how you stop cancer cells. You have to get rid of the cells that are doing the mitosis, which means you're putting toxins in your body to destroy cells. You target it so it hits the cancer, but you have collateral damage. Okay? They also have this nucleoid region. We don't really have that because we have a true nucleus. Their nucleoid region is basically where they have some proteins kind of supporting the DNA, but again, there's no like membrane around it. That's the big difference. Now, other structures that they can have as well. They can have what we call flagella. And the flagella are these little whip-like tails attached to their cells that help them swim through the environment. So they can find food or they can respond to different types of things like light or other bacteria they can swim through. Again, with the flagella, it may be a single flagellum or it may be multiple flagella. We have one cell in our body that has flagella. Does anybody know what it is? The sperm cell. If you ever get a chance to look at a sperm cell under a microscope, do it. They are so cute. They're just like swimming around. Sometimes you see one doing like donuts in the parking lot and you're like, okay, I hope that one never reaches an egg. Because it's just like, you're like, yeah, that wouldn't be good. I mean, that's the kid on the playground running around in circles and then falling down. You're like, yeah. Okay, that's so mean. Okay, anyway. So we do have a flagella, but our flagella is made different. We actually use different types of proteins and we use a different structure. So even though we have the same movement, it's a different structure pattern. Okay? It's kind of like comparing like a stucco home to a brick home. They're just a little different with how they're made. And then they also can have what are called pili. Pili are little short extensions that are shorter than flagella. These little pili are basically kind of like little spikes, picture like spikes, that help them hold on to substances. A lot of our pond water have pili, so they can hold on to substances. They can also build pili. They can make kind of like an extending pili that allows them to exchange genetics. This is a whole other thing we run into. Let's say I have a cell right here that knows how to make a capsule. Okay? Remember, capsules make the bacterium harder to treat. And then I have this little bacteria here who should have listened to his mother and stayed over away, but it goes walking on the dark side and runs into one of these cells that knows how to make a capsule. They will somehow interact. We're still trying to figure out how they talk to each other. But they interact, and somehow this one with the capsule realizes that this one doesn't know how to make it. And it builds a pilus. It goes, boop, 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 and it attaches to that cell, and it exchanges the genetic information, and now when it's done and that pilus falls apart, they both know how to make capsules. This is crazy. This is how we get antibiotic resistance. We have a bacterium that learns how to become resistant, and it bumps into its neighbors, and it goes, hey, guess what I learned? I'll share it with you. Yeah, and they share the information. They're like, yes, it's like sharing recipes, right? They're like, here, this is what you, okay, I know how to do it. So this is why we run into trouble with our bacteria. They are very friendly with one another. Okay, so again, kind of a simple picture of a bacterial cell. We have the cytoplasm, that's that jelly-like substance inside. These little dots are the ribosomes. What's made at the ribosomes? Proteins, good. We have our private protein manufacturing. We have our DNA in the nucleoid region, kind of the central region held with proteins. Again, no sap or membrane around it. DNA's coiled, circular, small. We might have a capsule. We can have a cell wall. And then we have this flagella here that is able to help propel the cell. Or we can have some pili, which are these little hair-like extensions that are able to be on the outside of the cell and help them stick, or even help them and sometimes to exchange genetic material. Okay. Prokaryotes tend to be very small. We've already seen this. Remember our E. coli? My gosh, it was so tiny. They average about 0.2 to 1 micrometers as far as how wide they are, and then the length is about 2 to 8 micrometers. E. coli is about 2. That's why they're so hard to see. They're right at the edge of what we can see. Most of the bacteria out there are going to be what we call monomorphic. That means they have one shape. Mono meaning one, morphic meaning shape. So, for example, E. coli is always going to be rod-shaped. Bacillus is always going to be rod-shaped. Staphylococcus is always going to be coxy-shaped, circular. A few bacteria out there are what we call pliomorphic. Plio meaning that they are a little odd, and it's hard to call their shape. Like, look at this one. This is like a baseball bat. It's got like kind of a rounded edge, so you might go, okay, well there it's coxy, but then look, at the end, the tail is like a bacillus. So we just refer to those as pliomorphic bacteria because we really don't know what to call it. It's like a couple bacillus. Like, what do you say? You know, it's like, I don't know, it's like a combo. The Frankenstein. Our three basic shapes of bacteria then are going to be bacillus-shaped, coxy or coccus-shaped. Coccus is singular, coxy is plural. And then spiral-shaped. So, coxy, bacillus, and then spiral-shaped. And again, the cells are pretty much going to have the same shape every time, and most of them are going to be monomorphic. This is where our writing style really begins to become important. If you're taking an exam and you're telling me that there is something that is bacillus-shaped, remember, if it's a shape, it's not going to be underlined, and it will only be capitalized if it's at the beginning of a sentence. So be bacillus-shaped. If you're telling me you're talking about the organism Bacillus, then it needs to be capitalized because it's a genus and underlined if you're writing it on a test. So make sure you got those nomenclature rules dialed in because they will come back. Now we can have different arrangements with our shapes, and these arrangements kind of tell us what to expect our cells to look like. For example, we can have diplocoxy. Diplo, when you see di, you think of two. That means we have two cells together, okay? For example, gonorrhea is a diplocoxy cell. When I look at a smear with a gonorrheal cell present, I'm going to see little twins everywhere, little double cells everywhere. They just hang out like that. Streptococcus, when you hear the word streptococcus, we have a genus called Streptococcus. That means that I have circles in a chain. Think of like a pearl necklace or like a long chain. So when I tell you this is the genus Streptococcus, you can picture in your head, okay, that's like a chain of circles. We also can have tetrads. These are where the cells are still circular shaped, but they split in fours. And then we can get these weird cube formations. At the bottom, though, are the other ones that are most common, which are the Staphylococcus. So if you're studying these, the ones that I really want you to know are the Diplococcus, the Streptococcus, and the Staphylococcus. I put the tetra and the sarcinae up there just because I want you to know that there are other arrangements, but we don't see those in here. So when you're studying, you can kind of ignore those. All right, so focus on the diplo, the strepto, and the staplo, because we're going to see them. We also can take that same logic and apply it to bacillus. You can have diplobacillus. Cool, me too, I know what that means. I can have streptobacilli. Great, that means chains. See, I can just like copy this over, this is great. I can have coccobacilli, that's where I have kind of an oval shaped cell. Again, not too common, so I wouldn't focus on coccobacilli. I can also have palisades. Palisades are where my bacillus, instead of growing end to end, grows side to side. They look like little picket fences. They're so cute. When you look at them on a cell, it's like a little picket fence or like a little defense lineup for a football team. They're all lined up. They're really cute. So palisades are something that are unique to the bacilli. So they're good to know because it's a unique structure. If I say which cells can make a palisade arrangement, they would be the bacillus-shaped ones. Okay? And then we have our spirals. Our spirals come in a vibrio shape. Think of a vibrio as kind of like a boomerang. It's a cell that just has a single bend to it. They can come in a spiral shape. These have come in almost like a helical twist to them. So they're kind of like a little spring twist. And then if they get really long and helical, I can refer to those as spirochetes. So the spiral shape, it's still fairly short and it looks like it has a little twist. A spirochete is where I'm seeing a long coil. Remember our syphilis that I said looked like little snakes? That's a spirochete. It was pretty long and it had a whole bunch of coils in it. So we'll look at more pictures of syphilis as we move along. So as far as these go, the ones we're going to see in here, we'll see vibrios. We'll see kind of a boomerang shape. And we'll see spirochetes. Those are going to be our two main ones that we really focus on. Okay, I observe a bacterium that is a streptococci. Everybody hold your hand up. Draw what a streptococci would look like in the air. Good. Excellent. Don't look at me like that. Yeah, like this, right? You guys are looking at me like, what is she doing? Yeah, we're doing air drawings of bacteria. When you're studying, you can do your air drawing. Excellent. Good job. You observe some gram-negative diplobacilli bacteria. What color would these be? Okay, they're gram-negative. What color are they?
Pink.
Pink. What would their morphology be? Morphology meaning shape. Two and bacilli, so they're rods, right? So like two rods. This is like learning another language. Can you imagine on the first day of class if I said, today we're going to look at diplobacilli bacteria, we're going to look at their morphology and whether or not they're gram-negative or gram-positive. You would have looked at me and been like, I'm going to leave. I'm going to go to a different class. But now look, you're understanding this. I'm throwing terms at you and you got it. This is cool. So the reason I tell you that, give yourself time and grace when you're studying. When you're learning microbiology, you are learning a new language. Just like it's hard for us to learn Spanish or French or Portuguese or whatever we're studying, it is hard to get these concepts down. Give yourself the time and grace to do it. Okay, I hear a lot of students go, I can't get it. Don't worry about it. It takes time. I'm still trying to speak Spanish. I've taken Spanish classes and I'm still bad at it. I can somewhat understand it, but I try to speak it. It's horrible. I can't conjugate verbs to save my life. They're horrible. You know, and I can tell you cool things like there's a mouse on the table, but that's not helpful. That's not helpful. That's what I learned in my Spanish class. You know, but that's not helpful. Okay, but the idea is that you have to give yourself time to learn. Okay, we can do a few more slides now. So let's look at a little more structure of that prokaryote. Let's go a little deeper. In our prokaryotic cell, let's start with the most outer layer we can start with, and then we'll work our way in. We're going to start with the most outer structure, which is the glycocalyx, and then we're going to talk about other structures that are found outside of the cell, like the flagella and the pili and the imbriate. Okay, so let's start with the glycocalyx. The glycocalyx is going to be this kind of sugar coat that some bacteria are going to be able to develop, okay? This sugar coat is made of a polysaccharide, so many sugars, and it may also have some polypeptides, some protein thrown in. If this sugar coat is attached directly to the cell wall, we refer to it as a capsule. So we saw pictures of a capsule earlier. Sometimes, though, this layer is not attached 100% to the cell wall, but just in a few little areas. It's almost like it's welded in a couple spots. That means it's not as organized, so we refer to that as a slime layer. That just means when I look at the cell, instead of having a nice rigid capsule right around the cell wall, it's more like kind of loosey-goosey. It's attached in a few spots. Now, the glycocalyx is going to help with virulence. It's like an extra shield or an extra protection. Remember, virulence means ability to cause illness and harder to treat, so it's more virulent. It also, because it's a little sticky, can help the cells stick to surfaces. So for example, if I have a cell come into my body with glycocalyx on it, it can adhere or stick to my cells or other surfaces in the body. Now, gram-negatives, they're always going to have some sort of a glycocalyx or a structure around them. We refer to that as an EPS, or an extracellular polymeric substance. Good Lord, this is what I'm talking about with a whole different language. It means that I have some sort of coat on top of my cell wall. Remember, our gram-negatives, their cell walls are so thin, so they're always going to have some other coat on top of that thin gram cell wall, so that they have a little extra protection, okay? And we'll look at pictures of them as we go through. So our capsule then, as I said, is attached everywhere to that cell wall, so it's very organized, very rigid. And what's going to happen is these capsules make our cells appear bigger. It makes them harder for antibiotics to penetrate. And it's going to do something where it's going to prevent phagocytosis. Phagocytosis means cellular consuming or cellular eating. The way this works, if I have one of my immune system cells, one of my white blood cells, come up to a bacterium, it's going to eat and destroy that bacterium. In this case, I have a capsule though, so my bacterium appears big. So now when my little cell comes up, it's like, oh, it's too big. Like it can't, it's like, you give somebody an elephant and go, here, you can eat this. It's like, it's huge, right? I mean, why would you want to eat an elephant? But still, the idea is it's big. It's a big bite. It's like, or if you go to a hamburger joint, they give you like a hamburger that tall. Have you ever done that? And they give you something huge and you're like, how can I even eat this? It's too big. Deconstruct it. The same is true with our cells here. It's hard for our immune system to get into. I'll stop here. I'll finish this and stop here. The slime layer is where I said it's attached a little bit. It's more loosely arranged. And so in this case, I'm not so worried about protecting my cells from consumption. In other words, I'm not worried about another cell trying to eat me. Usually if I have a slime layer, I'm an organism that lives like in the environment, maybe in soil or water. And what I'm more worried about is dehydration. So this slime layer that's more loosey-goosey, it helps trap water inside. It helps prevent the cell from drying out. So we see this, for example, with a lot of our bacteria that are found in like water environments, because that way if they get into a situation where it's dry, they can live for a couple days before they dry out. And their hope is that the water will come back. Okay, we'll stop here. We'll pick up here with our next lecture, which will be on Wednesday. Don't forget, Monday's a holiday. Okay, I always remind people when holidays are coming in, because I was the student that would show up.